Hey, so it's time. So let's start the session. So welcome everybody to the break session for infectious autoimmune disease. And we're happy to have so many audience here. I think the room is a little bit isolated, but yeah, let's start discussions. And okay, again, people just came in much more to make this because of discussion time. So don't be shy. And okay, how we start? Maybe this session, Yuki Okada from Breakout University, Tokyo, Japan, and yeah.
I'm Ponpan Matangkasombut from Mahidol University, Bangkok, Thailand.
So the purpose of the session is some discussion or accelerate further collaborations. So I think most people have some interest in this topic. So do you guys have microphone also? Yeah. Oh, yeah. Maybe why not? Because there are not so many. Yeah, so there are many people here. So just briefly introduce yourself to just and especially, of course, why you're interested in this session or how. Just briefly, yeah. Name and
Hi, everyone. My name's Lucia Robson. I'm part of the HCA Executive Office. I'm here to help make sure the people online can have their views and comments heard.
thank you. And
I'm John Randolph, also from the HCA Executive Office. I'm here to try to recapture my former scientific life as a virologist.
Chloe Villani, part of the HCA OC. I actually coordinate the ecosystem, but relevant today, I coordinate the immune bio network. So definitely wanted to hear about the interest in different immune disease. And as part of my own private work, we study a range of autoimmune and infectious disease. So curious to hear about what's relevant in this part of the world.
okay. And you? Oh, it's you. Yeah.
Hi, Alex Sharland from Australia. So I work in transplantation, but there are certain parallels and overlaps between self-reactivity and alloreactivity.
Thank you.
Hello, I'm Andrew Kwok from the Chinese University of Hong Kong. So I'm actually a neurologist, and I actually previously worked on infectious diseases, so in COVID, but also more generally in sepsis. And now I'm thinking much more about autoimmunity. So it's nice to, think about two sides of the coin. So hopefully we'll get some good discussions.
Oh, nice.
Okay. Hello, my name is Ananya. I'm from Riken IMS, Japan. So I'm interested in the immune responses to infectious diseases. Previously, I was working on dengue, but right now I'm working on a COVID infection and also responses to vaccination. Thank you.
Okay. Yeah, okay.
Hello, my name is Hui. I'm from Vietnam, but now I'm a PhD student in Chulalongkorn University in Thailand. I'm work in a lab of clinical virology, and now I'm have a project about HCV with unresponse with direct antivirals. Yes.
Hi, I'm Korpan Bunnak from Sirat Hospital, Mahidol University. I'm working on viral immunology. Thanks.
Hi, everyone. Some of you maybe now have seen my face. I'm Monica from Indonesia, from Okada Institute for Nanotechnology. Actually, I don't work on specific disease, but the group I'm in name Genome Diversity and Disease. So we aim to see any disease in the Indonesia genetic diversity. Thank you.
Yes, we are just briefly introducing ourselves and also showing the interest to start how to be. So we are looking for you're interested and for the discussions.
Yeah.
Please.
My name is Srivalli, and I am a PhD student at Institute of Systems Immunology at Würzburg in Germany. And I am mostly working on cardiovascular diseases. Thank you.
Hi, I'm Tibu. I'm working on host immune responses to the viruses.
Hi, I'm Damita, and this is Nalita. We are from Ajahn Ponpan lab. So from Mahidol University, we are like research assistant and mainly like a lot of research related to Ajahn Ponpan.
Hi, I'm Ponpan Walawat from Mahidol University. I'm a faculty member working on stem cell biology and single cell omics. Thank you.
Hi, I'm Yu Lai. From HKUST, and I'm here a volunteer too. Yeah, and I'm also very interested in the autoimmune disease here.
Hi, I'm Hu Jiawei from the Hong Kong University of Science and Technology. I'm also a volunteer here to do scribe work, and I'm also very interested in immunology.
Hello, I'm Nicole from the Chinese University of Hong Kong. And I'm very interested in the autoimmune disease, so I just came here to learn something new.
Okay, is there anyone who wanna? Yeah, because we are just collecting their names and interests. So yeah, please.
Hello, everyone. I'm Shejuti Shaha from the Child Health Research Foundation in Bangladesh, and I'm a molecular microbiologist. I work in the intersection of infectious diseases and public health.
Hi, my name is Yogesh. I'm also from the Child Health Research Foundation. My focus is pediatric infectious diseases, especially pneumonia and respiratory diseases.
Okay, good. So, oh, here's Kian Hong. He's a very famous guy. He's a leader, right? Okay, so maybe thank you for everyone briefly introduce yourself. So I again impressed by we have so many people from diverse countries. So it is unusual because most of the meeting were a small number of country, but many number from small country. But here we have so many diverse. It's very nice. And also I heard that you're interested on also diverse in many kinds of disease, infectious disease, more biological in response. And I'm very glad to have it. So maybe can you show our slide? Okay, so I and Ponpan had similar breakout session last year, and this is somehow based on what we discussed last year, and we wanna also show some updates. So yes, style aim is Pan-Asian health promotion control infections and autoimmune diseases, and yes, last year we have some discussions on this, and what is now in colored in blue underbirds, but we are after the discussion with last year, I and Ponpan has initially started, but again, the purpose of this breakout session is expand network or expand disease area. So maybe we are going to briefly introduce what we have been done last year, but also please also make your feedback so we just. So I remember we set a short-term goal. Anyway, we need to set some project, data, and funding. So we discussed what is prioritization of target infectious autoimmune disease. Because last year, the COVID-19 was one of the most interesting public health. Actually, we have discussion more on infectious diseases. And here comes so many disease candidates from the audience, such as tuberculosis, non-tuberculous mycobacteria, yes, NTM, right? Dengue, liver cirrhosis, and yes, malaria. And I think influenza is still getting again, but some after discussion, maybe some infection. It's a public health problem in Asian area. I think the prioritization disease, and my group has them also working in NTM, and Ponpan group is also working in dengue. Yes. And yes, also what is important, what is the target tissue of single cell mixed analysis? It always happens, right? More, of course, each researcher can access their own surgical samples, but yes, when we have on the international collaboration, which tissue to target is also very important. So peripheral blood, but skin, lung, bar, and but yes, anyway, peripheral blood is currently, I think, one of the most accessible tissue with equity so many across many agents, so that the people can more easily to join. Yeah, okay, what is the technical method of single omics? So yes, again, single cell RNA sequence is one of the best because also helps by 10x. I think the quality and also accessibility to this technology is more getting wider and wider in that series. So I remember 2 or 3 years before this session, many posters said, we started single cell, right? We're starting to make others. But I think this year we show so many posters. Doing single cell sequence itself is just a tool in science, and others has been already completed. So anyway, so we need more on biology. But anyway, single cell sequence is very. But ATAC-seq, Marchon, long-read-seq is, I think, is an emerging technology. Yes, Marchon, we still need some high quality in the variability of cells, and long-read with some technique to make a library. But again, expanding to this technology is very important. And yes, and also, yes, what is important is even when we doing, especially infectious diseases, what is the strain of the infectious pathogens is very important, right? And even with the tuberculosis, they have different types of strain, in each difference in poison. So definition or differentiation of these strain of pathogens is very important when we are doing it. And it's also then, because we are also shifting from single cell to population level single cell. And population level single cell, we should definitely consider diversity in human genetics. So integration with human genetics, or yes, it's very important, yes. So GBAS and EQTL, and how the GBAS on disease is related to EQTL of the disease and single cell transcript of the patient, very important. And I'm sorry, I skipped the five. Yes, everyone can now have single cell sequence data, but how to analysis is still different groups. Of course, transcriptome data, differential gene expression, everyone do, but I found many nice posters who are developing deeply into the part of the single cell data and get further information. And sometimes in TCR and BCR or more integrative stuff. And again, grant proposal is very important, especially to help or to improve or to promote equity in data access, or maybe equity in global foundation is very important. And so this is short term. And then after discussion with Ponpan, our group for NTM, dengue, and we are going to show some preliminary results for the past. But again, this purpose of this session is not showing the result, but promoting further collaborations. And also midterm goal is construct pan-Asian network and catalog within diversity. So this is I think, discussion item again this year. So from now, we are going to show some part of our progress in last year. But during we are talking, please thinking each of you have your opinion, right? Your interest or you requests on this point. And after presentation, we come back to this and then let's further discuss. Okay, so let's briefly discuss what we have been we're interested in. Yes, our group has been working on, yes, NTM, non-tuberculous microbacteria, something like MSC or cancer apps. And yes, it's a kind of the infection, not tuberculosis, and not bacteria, but mycobacteria, but it still have a health problem. And it's also within many soils and cells. It's always threatened our health, and not strongly infectious for healthy people. But when you get sick or when you get old, these are very strong problem, especially language diseases. And it's NTM studies always discussed in parallel with TB, and because these two are very similar and sometimes difficult to distinguish it. And again, our focus, our public health focus is not only seeing the TB, but also looking for NTM is still important to conquer these two diseases. Because also optimize the treatment of the cases. So there are two major types of the approaches to this NMS. One is GBAS. So again, this is first GBAS, not so super strong signal, but we found that also similar to other diseases, such as TB or COVID-19, host human genetics has effects on variability of TB. So we are now starting to, these are small GBAS, but expanding to much further pan-Asian way to expand these Mark Jibas, so Japan and Korea and Taiwan. And anyway, population level Jibas result is also now very important tool to decipher the pathology. But not only Jibas, but we also need to single sequencing. So last 1 year, we have conducted the initial single cell, and we have now started to get the pilot phases of the data. So because NTM itself is strongly related to T cells, so not only PBMC from the NTM patients, we put some stimulation statuses. But anyway, we are just starting to get this data and integrate how to integrate this data with GBAS. So overview, maybe we are now expanding collaboration, not from Japan to Korea and Taiwan, and going to expand GBAS and seeing how to, yes. And also, yes, pathogen sequence is very important. So there are so many strains. They have some difference in host genetics and single cell symptoms. As we are now expanding. So yes, again, I think this is one of the initial pilot study, but if you have very interest or further collaboration or insights, you have sample base, please contact us. Okay, this is a presentation from our side. And then, yeah, Ponpan, please.
Thank you, Yuki. I actually have only just one slide, and I would like to have this session open, and we have some more discussion from the audience. So thank Yuki a lot for a very nice summary on the first slide, and we're gonna go back to that and pitch out and get some of your ideas and your thought on each of those topics and which disease you would like to work with and what tissues are feasible and you like to work with what techniques and so on and so forth. And also what are the challenges that you're facing? What are the unmet need? And then lastly, what can we do together as a community to move things forward, not only for us, but for the whole community. Together, how can we help each other to move science forward in a diversity way, in an equity way, not inequity, right? So I work for quite a long time on dengue virus infection, and this is my only slide. One thing is because I will be presenting the dengue work in the afternoon session. So dengue is a global burden, and from this meeting, I've discussed with friends from not only us in Thailand here, but from Sri Lanka, Bangladesh, Indonesia, also have the same problem. It's a big global problem, especially in the tropical area near the equator. It's caused by dengue viruses, and it affects like 4 million people per year, and its incidence is increasing and the severity is increasing. There's no antiviral, and there's only 2 vaccines available, and the vaccine's not the best just yet, just because it cannot elicit proper immune response to across all 4 serotypes equally. So much more works need to be done on this area. So we think single cell RNA-seq is, and also spatial omics and related technology that we're talking about in this meeting is very instrumental in discovering the understanding, the in-depth understanding of dengue immune response, as well as potentially human genetics, EQTLs, and also host viral interaction, viral sequencing and host viral interaction, for example. So we also discussed earlier in the IDA workshop just before HCA Asia that not only in Asia, but also dengue is also problems in Africa and South America. So potentially this could be a work that's done across different regional network as well, not only focusing on Asia. So that's some of the thought that we came up earlier during the meeting. So that's all for my single slide. Now I just want to open the floor and hear your thought and what you're interested in and what you want to work on, from what disease you want to work on. Yuki is interested in NTM. I'm interested in dengue, but we're not closed. We're quite open for more disease to work on. Or maybe
what tissue? We're working on peripheral blood, but perhaps skin is interesting. Colon nasal swab, nasal biopsy might be very important for certain diseases, such as other respiratory viruses. What technical methods we can use? Of course, we were saying, okay, everyone can do single cell, but not actually everyone can do single cell, even though we think it's in the community, we think it's quite standard for doing single cell, right? And the field start to move forward to spatial, but that's not true for every part of the world. So in developing country, in low middle income country, we still have access issue on this, and how can we help each other to move things along? Right, we will have this amazing technology to help public health issues, not only in developed country, but also in a place where it's most needed, right? So besides technology, how can maybe look at, maybe some of you are interested in the pathogen side. I know I see poster from you yesterday that you look also at the virus side and at the antigen side that's closely interact with the host, right? And maybe if focusing on disease is quite difficult, and maybe each country has different disease of interest, another way we can work together, as we discussed in the year before, is to developing tools or integrative platform so that everyone can use, and maybe from pathogen to host genetics, host immune response, pathogen genetics, and the interaction. And somehow we come together and we have this platform. If you're interested in dengue, you can use. If you're interested in NTM, flu, COVID, maybe that's one way we can go about as a community. Or, and also last things that Yuki mentioned is using population genetics together with the single cell transcriptomic data to provide us some more useful and a better in-depth understanding. So, okay, so that's all the 6 things that we want to open up the floor to discuss. And I start to see some hands up, and please let us know what disease you want to work with, what tissues, techniques, and platforms.
Hi, so I'm Tibu from Sri Lanka. So as you also interested in dengue, I want to just ask, because yesterday also we had a quite good discussion. So as we all know, the total dengue cases are around like 400 million, but it's reported only around 150 to 200. So that is basically asymptomatic dengue is there. But nowadays, this symptomatic dengue is increasing because of the comorbidities, because we have seen that the comorbidities are related to the severity of disease, well documented in COVID, because it was all around the world, so it's very well documented. So now these symptomatic dengue are increasing, and we have seen like plasmablast expansion and non-neutralizing antibodies and all. So for a developing country like Sri Lanka, if the single cell is not quite possible for, as it's expensive, so is there any other possible way to study about this severity which is related to comorbidities? Because now it's been seen increasingly when it comes to Sri Lanka. So like what it would be possible to study about that and the severity, and because we are seeing so many adult population are turning up to the hospitals with the symptomatic, and they are admitted to like ICUs, and we have been seeing some mortality as well. So is there any suggestions and anything?
Max, because I have questions back to you. Do we have 2 mics? No?
Only 2.
Okay. I'm going to go next to you, so this is only partially awkward. We can speak next to each other so that the moderators, kind of a little duo on this side. So before I ask you back some question, I guess a bigger comment for the discussion. You saw a QR code yesterday. It was tied to spatial, but globally speaking, this is more about defining disease because as part of our next phase for HCA, we want to try to figure out which global disease burden we could help prioritize so that we can help raise awareness and fundraise and tackle problems like the ones you're pointing out. So it sure looks like the dengue fever, this map definitely helps push the message of the global disease burden. So when you see the QR code again, it's not necessarily means it's not just for spatial. It's about defining which disease priority you want to do, accounting for the fact there's a lot of other disease consortium. We want to distinguish ourselves by focusing on those that might be neglected. And I'm not talking about neglected disease, but more those that are hard to study. So keep that in mind. So dengue, good one. You said it's hard to generate data. Are you capable of banking samples to start with?
Yes. So when it comes to Sri Lanka, so we are University of Sri Javadhanapura. We have our like teaching hospital called Colombo South Teaching Hospital. So we have access to the patients, and also we are closely working with the National Institute of Infectious Disease. So we are accessible to the samples, and we also have some PBMCs which we have from like last 3-4 years. So since COVID times and before COVID as well.
So you're capable of banking PBMCs and you can store them at -80.
So we are doing the cryopreservation. So in the liquid nitrogen and -80s,
And
yes.
do you keep just making sure there's the problem of generating data, but the first problem is to bank the data, bank the sample. So you can bank the PBMCs, you can bank the plasma, and right?
Yeah.
Okay. Well, that's a first. In terms of solution, I'm not the one that's going to solve the solutions for measurements, but there's definitely different platforms that are emerging for single cell suspension that goes beyond the next genomics that can be considered. He's hiding on the far right, not pointing him because of funders, but CZI has done this great partnerships with Scale Bio and NVIDIA, for example. There's a program that John Nicole mentioned yesterday for the 100 million cells. It's not the last program. So I think as we try to organize a community to define, who has samples, what are the big disease burden, can we identify? So we have, in Sri Lanka, here in Thailand, maybe we can identify some growths in South America and really leverage the global reach of HCA to try to kind of bring it all together and make it more compelling. So that's just that's one of the things that should come up from this type of effort. That's one that goes beyond this. The second question I'm going to ask you said how it's a big burden in the hospital, but beyond profiling, I think it's important to define what questions you want to answer to analyze some single cell. What are you trying to do with single cell that you cannot do with other cost-effective measurements? What's the question being asked?
So the thing is we want to answer is we have done basic immune response studies, how these things working in the pediatric population or adult, and how so they're supposed to we have done in our lab so far, which is not genomics but mostly immune experiments. So what we want to answer is because if you see the population, the symptomatic cases and the severe cases are increasing. So as Sri Lanka having a good health system, we are somewhat managing, but it's overwhelming the health system. So we wanted to study why this severity is increasing. One relation only we found is when we have this cohort, which longitudinal cohort we have, that this BMI and diabetes are keep on increasing in that particular study area. So we wanted to address that because this metabolic disease is not only a problem in Sri Lanka, but also all over the world. And also we are seeing the expansion of dengue into the Europe as well. So it would not be only to the tropical region, but it also in future, it would help the other regions as well. So yeah.
In terms of understanding the disease risk that are tied to comorbidity, and if you can predict who's at risk for more severe complication is your question.
Yes.
And understanding the biology tied to severe complication could help you think about predictors, but also therapeutics. That's kind of the idea.
Yeah. So because For example, B cells or T cells, like how it's going to be here.
Yeah.
So
I just sorry, I didn't mean you're the co-chair. I think like framing these questions so that you can connect, go beyond. If you also go beyond Asia, that makes an even stronger case for HCA to kind of help support this matchmaking and kind of make it priority. But so also as you think about these, I think it's helpful to define the questions you're trying to answer.
It's great. Thanks a lot, Chloe. I think that's a very important issues and for the access to the technology and maybe some program like Scale Bioscience can help alleviate that. But there's also some challenge on this front in that in certain countries, samples cannot be shipped out to other countries. You can add on to that.
I think we should I mean, you're right. It is true that with the current offering that was with shipping, and I guess I'm not suggesting that the solution should be shipping. The first thing is locating who has samples or who can collect samples, making sure to collect them the right way to start with, and that you agree on your metadata information for patient. Second, and then when as we kind of figure out, I'm not doing any commitment, I'm just thinking out loud. And so as we figure out who has cohorts, who's building effort, there might be an opportunity to think about center of excellence, but where we can help develop some local expertise that's closer to your region potentially. So understanding who's interested, what capabilities you have locally, what's the closest capabilities. So because that also helps in terms of doing cost mitigation, as opposed to having to ship it to your lab, depending on where you are. If we can help figure out where are the hotspot of experts and interest. I realize like shipping samples outside your country has all of these different legal complications, which is not necessarily a solution. Sorry, I wanted to clarify.
No, that's great. That's great. So maybe regional hub is something we can like. Yeah. Sure. Cool.
Oh, yeah, so I just wanted to add this to what my friend Tibu said. So in terms of what questions we're trying to answer with single cell in terms of dengue and also tuberculosis, which is actually the prevalence is increasing in our country. One, due to the increasing prevalence in HIV, and also because of certain socioeconomic changes that have happened recently, the nutrition has reduced drastically. And there was a paper out, I think, by some community physicians actually suggesting that could be a correlation. So we're seeing TB rise. And so I'm also a clinician as well as an immunologist. And a question we get from clinicians managing dengue and TB is, do you have a biomarker to see who's going into, say, paradoxical TB, which is a huge clinical issue? How can we predict that? And same with dengue, because we've been managing dengue for so long, we still don't have like a biomarker which can like kind of differentiate, is someone going to be severe? So we, as doctors, you have to kind of monitor everyone, which is very labor intensive. And that is a question people have been asking us, we've been asking ourselves, and we're hoping maybe having a little bit more insight with into like a wide array of genes could all help answer that question, and maybe drug targets as well. Thank you.
Yeah. Oh, yes.
Okay. Is that on now? Or, I don't know. I'll speak loudly anyway. So okay. So you were mentioning that the vaccine responses to the dengue vaccines were variable and not necessarily uniform across all serotypes. And I wondered, forgive my ignorance here, I don't know whether AIDA covers immunoglobulin germline analysis, because that can certainly influence the vaccine response and whether you can connect those together. I guess the other connection I was thinking of is, as climate change takes hold and parts of the world that are not currently affected by dengue, as our colleagues said, become affected or become susceptible to being affected? Is it possible to leverage that for funding?
Okay, very nice. So maybe first comments for the vaccination response. I fully agree. Now we have we can see some reports on genetics on vaccine respiration, mostly on COVID-19 vaccine, but we found that most of the regions, something like GH, MEC, also reported that vaccination response with other pathogens. So it looks like some specific genetic locus is strongly regulating acquired immunity in humans. So vaccination result in one specific pathogen can be extensionable for other disease. So in some sense, I think first, we have so many resources for COVID-19 vaccine, but these results can be also helpful when you design or thinking of other vaccinations. So this is my first comment. Okay, and quickly, the second comment.
The second comment was whether, the susceptibility of parts of the world outside the current range of dengue to being next in line might prompt increased access to funding.
So we're actually try to leverage that. So back in 2010, the European Union actually have concern because they have the mosquito vector, Aedes albopictus, in the area, and they started to identify some hotspot from dengue infection in Southern Europe. So we actually got funding from the European Union, FP7, to conduct the cohort, which I'll be presenting later on this afternoon. So I think that's a very good idea, and maybe we should not stop there, and we should try to encourage some more, because global warming is an ongoing process, and it has been clearly shown that mosquito vector residence has expanded away from the equator because of global warming. So excellent point. Thank you. And maybe I'll touch upon a little bit on the AIDA immune repertoire. So JP team, Jung-Un Park team, is currently analyzing the BCR, TCR data of the AIDA projects. Not quite done yet, but hopefully soon. But it's a very important point that immune repertoire in different population might affect the immune response to vaccine, right? And also the vaccine design is also crucial. It's like interact with each other. So both the antibody response and also T-cell response, I'm particularly very interested in the T-cell side. And so that HLA in different population is quite varied. So how we design vaccine to cover like the global population would also be depends on the HLA type and the human genetics across all population. I think that's a very important point, and there's a lot of work need to be done regarding this. Thank you. Judy.
Loving all these discussions and conversations on dengue, one of my favorite, bad word, but favorite diseases, also very high burden in Bangladesh, and we have cohorts that we're following. But I wanted to kind of perhaps bring the room back to like infectious diseases as a whole and prioritization. A lot of investments in the last 2 decades have been put into prioritizing diseases. So WHO does ridiculous amount of work prioritizing pathogens. There are committees, there are advisory boards, there's a lot of funding in just figuring out what diseases to prioritize on, because you can imagine industries, let's say, what should they invest on? What vaccines are next? What antibiotics or antivirals are next, right? So there are actually these studies done, very, very systematic studies where WHO or commissioned groups interact with each of the regions of the world and come up with this priority list of pathogens. And every few years, this priority list of pathogens are revised. It even comes up with like why the WHO even comes up with why these diseases are important, what are the gaps in data at the moment, and what do we need to do to prevent or treat these diseases. So in 2024, the new list was released, and for the first time, WHO has included dengue in it. I think that is something we could also focus on. What are already the diseases that has been prioritized, and all global donors for infectious diseases kind of look at that list, right? So Gates Foundation will actually change its priorities based on, modify its priorities based on what are the priority list and, Wellcome Trust, NIH, NIHR, they will all look at that priority list and figure out what to fund. So maybe that is something we could also revisit. Otherwise, we'll just be always interested in our favorite disease.
That's right. That's right. Thank you. Thank you, Sanchuti. And I think HCA also moving forward, like in the general meeting, right? John can give me some more advice and chime into this. So I understand that in HCA general meeting this year, HCA plan to move forward to more disease oriented and also look at this list of priority list, right? So would you like to make some comments on like priority list focusing on infectious and autoimmune disease?
I'll make an initial comment. I don't know, Chloe, you might want to add something too. So first of all, that's actually a big part of why we implemented this survey that we're piloting actually at this meeting. And so if you have not taken that survey yet, and maybe we can work on putting the QR code up at the end or something, I don't know. But please do take that survey because that really will inform what we do in the path forward. I think there's also an intersection between public health interest, opportunity. I mean, you'll see on the survey that we ask what you think should be the priorities in your region, but then we also ask what the funders' priorities currently are as well. And the question is there a mismatch, or and is that an opportunity for education, or if that's actually aligned, that might really be an opportunity to think about a program in that disease of interest. So it's just to say that this is very early for HCA in this process, but we're really serious about getting feedback from you and hearing where, not just what you think should be the priorities, but where you really think the near-term opportunities are, where the samples already exist or could be generated very quickly, and so forth. So please do, we're very serious about wanting input on this. Do you want to say anything more? Or?
You summarized it beautifully. We also talked a lot when we're starting to, and I couldn't agree more that this is very early, right? We're trying to do information gathering because it doesn't mean that it should be only one effort, let's say just dengue. But to your point, actually, we're like, well, we need to look at DALIs. We need to look at what WHO is prioritizing because that will help in tying with funding, but also to define the gaps when there's no, there's some neglected disease, and if it matched with some of the cohorts we're building, some of the local disease burden. So we're thinking about global disease burden. That's would be a good one with the map you were showing. When you were talking about dengue and climate change, I was looking at how many cases were reported in North America, and I was like, wow, there was close to 10,000 cases this year. And so that's an example, but it might be things that are more very local to your region. And so local could be local to your countries, but that could be an opportunity to find partnerships with local governments. So we want to hear about the current problems you're interested in, the type of court you're building. So it was very helpful to know that actually, while you cannot generate data, actually you can bank samples, you can get information. That's the type of information we want to know, because it means that if we found different partnerships for funding, you would be ready to go. Right. I mean, I realize you may not be ready to go tomorrow because we need to develop the local expertise and bring the funding, but that is like getting the patient specimen is not a trivial thing to do. And so if you already have that, it means you have some forms of consent form, some forms of IRB approval. The inertia may not be so big to kind of get things going. So we want to use a data-driven approach to kind of figure out what's of interest and also to make sure that we don't reinvent the wheel, we don't overlap. There's a lot of, in Europe and North America, a lot of different disease consortium is being well done. The idea is to not overlap. There's too many questions to be answered. So I was very curious to hear about what you're interested in. It's also interesting that, there's comorbidities between both of your interests, which is also extremely relevant in trying to think how we can build effort that really synergize. So that's actually extremely relevant that, there's some overlap between what you're interested in. And so yes, do fill out that questionnaire. We're gonna remind you many, many, many times, and you're doing yourself a favor by letting us know, because we cannot read your mind. But yes, I was looking at the WHO report when you were talking, and so I don't think I found the right one because I couldn't find dengue, but I saw Zika, Lassa virus. And so the other thing I was going to say about dengue and severity is that COVID may teach us some of the lessons that could help in terms of defining the biology of host response and severity that could be leveraged. And how many cells are needed to answer questions, at what type of genetic information, what type of other measurements. You're asking about biomarkers. It's absolutely not cost effective to do single cell genomics, clinically speaking. But of course, one can define a specific genes, a specific protein that could be done. And I think there might be a way of having some lesson learned in terms of cohort size, number of cells, number of measurements, because there's a lot of data that were generated to some extent.
Yes, thank you. I have some quick comments on the banking resource. Yeah, I fully agree. Banking is really important. So how to collect sample is technical point in disease genomics. And maybe most of the disease, okay, define disease and collect sample for us. But I think infectious disease using the previously collected biobank resource is very useful. For example, asymptomatic cases, difficult to define asymptomatic infectious disease cases, but you have some biomarker, something like antibody to some specific antigen. Here's a biobank resource. We can retrospectively affect the populational impacts, how people with severe asymptomatic exist. So I think most of the disease, most of countries now starting to have some biobank staff or hospital, and just recheck these resources. You can more precisely define the degrees of infectious patients. Sometimes better than just starting recollect the sample by yourself. So, for example, especially the disease with common infectious rate. For example, in Japanese, pylori, right? Many people have pylori but don't know because they don't check. But you have serum in biobank. You can just check the titer of pylori antibody. You can qualify. So, retrospectively collecting the patients is very important. Maybe you also know the story EB virus or multiple sclerosis. This is defined by rechecking the EB infection status in, I think, veteran, medium veteran, or US Army cohorts. Yeah. So, and also this is also very important related to the biomarkers. We always see trying to find biomarkers for the affected patients or severe patients. Most of it they are just consequences but not causes. But you have data or samples who have before getting infection, right? So metabolized or some protein assay in such population level and can be a causative biomarker to accelerate severity. So in this case, your country have some certain number of resources, biospecimens. Rechecking these resources with clinical data or re-evaluating their seropositivity, it should be very helpful to define this point. So which we sometimes do in our biobank, and maybe we found some the patients who were not recognized, but this kind of patients should be in the certain amount of the populations.
Yeah, I am following what you're saying, and I guess I'm gonna add a little bit to what you're saying. I also agree that you need a broader cohort, and also there's always limited resources in terms of how many patients we can profile in terms of single cell multi-omics, and repertoire is actually really key in the context of infectious disease. And if you're going to try to translate this back to the clinic, some forms of surface protein is really important. So being able to find ways of identifying more severe patients is really key, and also figure out where exactly they were infected so that you can actually translate back to the clinic. It cannot be a catch-all. I'm just going to profile patients with disease X, And so as a community, decide what priorities you want to focus on. I think also agreeing on different type of measurements that are fairly standard that could be applied so that you all develop a common lexicon to do patient selection and be able to bring your data together and harmonize would be really important. And so, viral load, viral titer, like being able to kind of have other measurements that helps classify your patient in a meaningful way is really important.
Yeah, I also just think so viral titer can be now most of the cases estimated by whole genome sequence data. So human whole genome data includes shallow amount of virus, especially DNA virus things. And if you have whole genome sequence data, you can specify people who have associated with specific virus. I think we can screen 1,000 viruses. It is much faster than you did assay. I think UK Biobank is going to release the I think herpes virus infection status based on whole genome sequence. Anyway, existing data can help you to identify specify this disease more than we always expect.
Yes, but I mean, also thinking about like a cost-effective way, whether it's like qPCR, like a ELISA-based, like it needs to be something cost-effective so that people can actually do this at scale.
Yeah, exactly. Yeah.
I'm curious. I don't want to derail. This is very good conversation. I mean, I guess it's a lot of blood-based, which perhaps in the context of infectious disease is what makes the most sense. But you mentioned your session also had autoimmune disease, and it's totally okay if there's no interest in autoimmune disease. I did hear one person in their introduction who said autoimmune disease, and there's also some comorbidities sometimes when you have autoimmune disease. And I understand that when you have really high disease burden, infectious disease, autoimmune disease, maybe you're the least of your concern. But it was the name of your session, so I was just curious, wearing my hat of the Immune Bio Network.
I think if you put this question to Immune Society, everyone responds different. Sorry, it looks like for me, I have been working on autoimmune diseases, right? So genetic risk directionally, it's I think somehow different between infection disease and most of the case autoimmune disease. So different. For example, when we do the Mendelian randomization analysis from SLE, they are protective for COVID-19. But you know, if any kind of direction you can identify any kind of explanation, so interference. But I think yes, associate comorbidity or less comorbidity between autoimmune disease or immune-related disease, infectious disease, I think is a strong interest of the field. So when we do genetics, we can see somehow positive and negative genetic correlation, but further validation should be done omics analysis.
I think that one of the power of single cell genomics and spatial is to try to kind of move away from just genetic variants, because for autoimmune disease, most of the relative risk is extremely small and cannot be explained by single cell. So it's not like cellular therapy will solve your issue. So being able to use, single cell genomics to understand the more complex cellular ecosystem across population and different ancestry to try to understand if the cellular drivers of tissue damage and chronicity are the same or not, there's some power to it. So moving away from polygenic risk score and moving away from blood. But I'm not clear if within, this part of the world, this is a pressing priority or not, just to get a sense.
I would say that there are very few epidemiological studies that have looked at like autoimmunity, at least in Bangladesh's case. Anecdotally, we know that the rates of like IBD or non-alcoholic fatty liver are very high, like especially in like South Asian population, and IBD rates are increasing as diets are changing and as like environment itself is also changing. But the problem really has been the fact that, well, in infectious diseases, there has been a lot of funding to do surveillance studies and actually catalog the burden of these various diseases. In autoimmune diseases, that hasn't happened. And I feel like before, like maybe some people can look at like some broad cohorts that they're building on, but so far, like that kind of a data at least is not available so much for autoimmune, and at least I can say that from South Asia's like.
So would you say in this case that it's more like it's neglected because of funding opportunity, but there's still a local disease burden, simply is not studied because of lack of opportunities?
Yeah, I think it's just the fact that the rate of so like infectious disease burden is coming down and the chronic disease burden is going up. But I feel like the health system and definitely not the research ecosystem hasn't been able to make that switch, because it's not like, although the infectious disease burden is going down, it's not going down. It hasn't gone down enough for the switch to occur. And so I feel like it's caught in a place where everybody has a sense of or understanding that like autoimmune disease are really important. But I feel like before, like, the burden data is not out, you can't quantify how much of a problem it is, or you can't you can't actually begin to do these like very targeted analysis. But I mean that needs to change, I guess. But I can't, like, I would say that epidemiologically, like, I think we can't define it yet.
But in terms of medical diagnosis and access to treatment, do you still do some forms of biopsy to diagnose patients?
Yeah. So for IBD, I mean, there are at least in Bangladesh, like there are like for like doctors and clinicians, they have an understanding of IBD. There are also certain drugs, not antibody drugs, but like tofacitinib and a few other of these like small molecules that are available in the market, and they're given to patients. So there is definitely like there's definitely burden there, but it's just not quantified.
That's super helpful. Thank you.
Yeah.
Maybe I chime in a bit from the Thailand side. So we also have burden in autoimmune disease, particularly like SLE. We have somehow have more severe disease in SLE in Thai patients, and some other certain autoimmune disease are actually more prevalent in our region as well. So that definitely patients out there and physicians who are interested in like learning more in research environment, but same problem with the funding. So it's a bit harder to get funding on autoimmune disease or allergic disease or something that's has been problem worldwide and not like very tropical, very highly burdened in the country. I think that's we share the same.
I just wanted to add something here, if I may. So yes, number one, oftentimes we think there is no burden because we're not specifically looking for it. The moment we'll start looking for it, we'll realize. But I think a great resource to understand burden are basically immigrant populations in different countries. And there we have really good data. For example, for a lot of the diseases, we get data from Bangladeshis living in the UK, Pakistanis living in the UK, because they have better access. Gujaratis, yeah, actually sometimes we get like more granular data from UK and US than from countries. So I think that would be a really good resource to look at. There are diseases where we completely find out from NHS of UK that what this existed in our population and they picked it up. So I think that would be a very good resource to look at.
A good point.
Good. So we are almost at the time. But is there anyone who wanna say message to the community? Anyone? Yep. Yeah. Please go.
Sorry, I'm not sure if this is too much of a pivot, but I don't actually work with human samples. I work a lot with ecology and mainly right now insects and plants. And because Ponpan, you mentioned that because of climate change, the vectors are spreading. So I'm wondering if more collaborations can be done between like, molecular biologists and entomologists and ecologists to like study the disease vectors and understand their like distribution and their life, like their habits and everything, so that I feel like this information from this kind of research might be valuable for like the prevention of the disease before it even happens in terms of infectious diseases. And as a ecologist, I want to say that we have very limited funding compared to you guys. So yeah. Okay.
You can just ask Shizetai. Yeah. Kihan, do you have any comments as a leader of HCA?
I guess maybe thinking about the last point on the list, different infection disease in different country, and if I may be permitted to do a bit of blue sky thinking. So Jay was talking yesterday about how disease is not categorical, it's continuous. And then also thinking about the way COVID-19 just affected every single country. So whether thinking about respiratory diseases and pandemic surveillance is something that could tie together every single country that's participating in the HCA. So if we, let's say, think about stuff like the Seattle flu study, which I heard of from Jay Shinduri in a talk, where they're doing community surveillance in Seattle for severe flu cases, and just try to have each country sample respiratory diseases as sort of a pilot study, then would that help prepare for future pandemics and tie together the public health need with single-cell genomics profiling? And maybe each individual country wouldn't have enough samples to be able to do that correlation between the pathogen and the immune readout. But if we're able to do this across all of the countries in the HCA and bring together all of the underrepresented countries that may not necessarily tie in with the disease focus we have over here. So I was wondering what this group thought about in terms of thinking about respiratory diseases as a bucket and surveillance to be able to bring together underrepresented communities beyond the folks that we have represented here.
Okay, so we almost at the time, so maybe wrap up the session. Maybe we're going to have a quick feedback later, but thank you again for everyone joining the break discussion. And please note that here's a nice community next, so maybe when the next pandemic, when this is something, this community will be good to proceed the collaboration together. Yes, Ponpan, please last remark.
Thanks a lot for the great, sorry, we're out of time, but maybe we can keep some continued discussion during the lunch break. But let me just summarize quite briefly. It's a very fruitful discussion. Thank you everyone for your participation. And I think we get some ideas, both the existing ideas, new ideas, and maybe we could try to work together in several ways, such as co-apply for funding from several countries or several regions, capacity building, establish local expertise team that maybe we can leverage and use that as a hub, and maybe establish platform and tools and we can help each other. So few announcement, we have lunch already there, lunchbox, the same place that we have yesterday. It's already set, so they remind me to finish so that you can have your lunch and not getting too hungry. And we'll have an infectious disease and autoimmune disease session again this afternoon. So if you're interested, please stay tuned. Thank you.